AP Biology, Unit 5 on heredity, and this is video 4. We're getting into non-Mendelian genetics. So before we get into non-Mendelian genetics, let's just remind ourselves what Mendelian genetics is all about. So Mendelian traits are those controlled by a single gene and have dominant and recessive alleles. That's helpful, it's useful as a starting point, but almost everything is non-Mendelian. So here we go. First example of non-Mendelian traits are polygenic traits. So remember that Mendelian traits are those controlled by a single gene. That's called monogenic, mono meaning one. Polygenic traits are those controlled by many genes, poly meaning many. So what you end up with when you have polygenic traits is a continuous sort of pattern of phenotypes. So you're not going to just have two different phenotypes, you're going to have a whole range. So one example of a polygenic trait is skin pigmentation. It's controlled by multiple genes. Skin pigmentation is an adaptation to the amount of UV exposure. It's independent of almost all other traits. So just from looking at someone's skin pigmentation, almost every single other trait about them, you just can't know. Something else to point out is skin pigmentation also does have to do with environmental influences in addition to genetics. We'll get back to environmental influences later. Another example of non-Mendelian genetics, codominance and incomplete dominance. An example of codominance are the A and B blood types, or alleles for A and B. And incomplete dominance, you can see on the right here, as the flower color. So see if you can define codominance and incomplete dominance using these diagrams to help you out. Codominance is where the phenotype of both alleles are exhibited in the heterozygote, and this is because both alleles affect the phenotype in separate distinguishable ways. So for example, in blood types, the type B and type A alleles are codominant because they both produce a unique antigen. You can see that in the sort of orange row of the diagram. Antigen A is shown as a little blue dot. Antigen B is shown as a sort of purple triangle. And so then if you have a copy of both A and B, instead of one winning out over the other, both of those antigens are produced. And you can see that in the diagram showing that both those blue circles and those purple triangles are produced. Now both of these blood types are dominant over the lowercase I blood type, which codes for O, type O blood. So if you have two copies of the recessive allele, that codes for type O blood, and that's because neither of the antigens will be produced. All right, incomplete dominance, on the other hand, is where we have the phenotype of the heterozygote is in between. So it's a blend of the phenotypes of the two homozygous. So you can see a homozygous dominant flower would be kind of that reddish color, and a homozygous recessive would be the white. When they breed together and produce that F1 generation, it produces an intermediate phenotype of those pink flowers. An example of incomplete dominance in humans is sickle cell. So sickle cell is controlled by a single gene, and there are two alleles. If you have two copies of the sickle cell allele, that will give you sickle cell disease, where your red blood cells are all a sort of shape that is kind of like a sickle. So a sickle is shown on the upper right there, and they don't carry oxygen as efficiently. If you have one copy of the sickle cell allele and one copy that doesn't code for sickle cell allele, that will give you sickle cell trait. So you will not have the effects of sickle cell disease, although there will be some symptoms when blood oxygen is very low. What I'd like you to do as practice is to draw a Punnett square showing the possible resulting offspring between a male and female who both have sickle cell trait. And to use in your Punnett square, since there's not a simple dominance recessive, we don't want to use capital and lowercase letters. So the common representation for the normal hemoglobin allele is HB superscript A, and for sickle cell it's HB superscript S. So see if you can try out a Punnett square. So when you do the Punnett square with two individuals that both have sickle cell trait, you'll find that a quarter of the offspring will be predicted to not have sickle cell disease or sickle cell trait. Half of the offspring will be carriers, will have the sickle cell trait, and then a quarter of the offspring will have sickle cell anemia and will be affected by that disease. So why is sickle cell trait an example of what we call a heterozygote advantage? Heterozygous advantage is where it's just what it sounds like, that there's some advantage in being a heterozygote. So there's an advantage in being a carrier. So in this case, individuals with one copy of the sickle cell allele are less susceptible to malaria. So the prevalence of sickle cell allele is most common in people whose ancestors come from areas where there's a high prevalence of malaria. You can see that in these pictures. So distribution of malaria is shown on the left, and the sickle cell allele is shown on the right. Because the sickle cell allele, HBS, is predominantly found in people whose ancestry comes from these areas of Africa where the prevalence of malaria is highest, there's a misconception that you can tell if somebody is more likely to have sickle cell based on their skin color. And that's problematic for a lot of reasons. So I want to take this opportunity to talk about the important difference between genetic ancestry and race, and why it's really important to not use race when you're talking about biology or when you're talking about medicine. So the first thing that I want you to understand is that compared to other species, humans really aren't very genetically diverse. Big takeaway is that all of us share way more similarities than differences. One way to see this is on the graph on the right. We can look at the relative differences between mitochondrial DNA within different species. And you can see humans are way, way down there towards the bottom. You see on the bottom right is humans. And so because humans are so far down on this graph, what that indicates is that humans actually have very few differences in their DNA within our species. Most other species are much more genetically diverse. The other things that I want you to understand, and there are 5 of them, so we're going to go through one at a time, and I should say all of these come from, I've listed the sources at the bottom. I would really recommend you do some additional reading on this. We'll have some class discussions, but the more you know about this, the better, and that'll really help us in our class discussions. But let's go through these 5 reasons why race is not the same thing as talking about human variation. So evolution rather than race is what explains human biological variation. Race as biology is based on the false idea of fixed, ideal, and unchanging types. Race categories were first a European folk idea from an era in which the world was seen as fixed and unchanging. All of that changed with evolutionary theory. So once we understood evolution, we have the tools to better explain human variation. We do not need to use outdated methods of understanding human variation. The illusion of unchanging racial types is completely incompatible with evolutionary theory. Human variation is continuous. So you can see that in the picture on the right, which shows human variation in height, which is another type of human variation. So there's no clear place to designate where one race begins and another ends. Skin color, for example, the physical characteristic we most often use to distinguish races, slowly, continuously changes from place to place and person to person. If one were to take a walk from Norway to Kenya, one would encounter slow and gradual changes in skin color. There's no place to unambiguously say where dark skin ends and lighter tones begin. Same is true about individual variation within groups. Line a group of individuals up by height and one can see that variation in height is continuous. Drawing lines separating groups is completely arbitrary when it comes to race. Okay, the third reason that I want you to understand of why race does not explain human variation is that human biological variation involves many traits that typically vary independently. Skin color, for example, is only correlated with a few other traits, such as hair and eye color, leaving unpredictable the huge number of other traits. So we'll talk about the mechanism of linked genes later on in this video, but please understand that almost all traits are completely independent of skin pigmentation. So thus it is a truism that race is only skin deep. So to get a visual example of this, imagine there are 4 traits represented in 4 layers. In this case, suppose that skin color is on top, and think of these 4 cores as either individuals or groups. The 2 on the right both have light skin color, but differ in all those other traits. This is because the top layer skin color does not predict the variation in the other traits or layers. Skin color is independent of almost all other traits. And this visual diagram I think is very helpful, but I hope you also understand, due to Mendel's law of independent assortment, why, like the actual mechanisms of meiosis, of why multiple traits don't have anything to do with each other. Number 4 that I want you to understand is genetic variation within so-called races is much greater than the variation among them. So for example, two individuals who identify as white might well be far more genetically different from one another than from someone self-identified as black. Any two humans share 99.9% of their DNA. Of that 0.1% that is variable, 95.7% is due to variation found within population groups. So the few traits that do vary more between populations than within is skin color, and we know the evolutionary history of that. It doesn't have to do with race, it has to do with UV exposure. And finally, number 5, there's no way to consistently classify human beings by race. Race groups are impossible to define in a stable and universal way. Nobody can agree on this. Nobody can agree over time. And so if one cannot define groups, one cannot make scientific generalizations about them. So for theoretical reasons, practical reasons, and scientific reasons, race fails to explain or describe human variation. I do want to make sure that you understand that I'm not saying that race doesn't exist. I'm just saying it's not a biological construct. It is a social construct. And so it certainly does have an effect on people's lives, but it doesn't have a biological meaning. But let's take a look at where we do have an intersection between race and something related to science, which is medicine. So in medicine, it's been common to list race as a risk factor. A risk factor is just one of the causes of a particular disease or medical condition. So for example, race has often been used to associate with birth weight. Birth weight is important because it's the main risk factor and predictor of perinatal and neonatal mortality. For decades, the average birth weight of black infants consistently measured at nearly a pound less than of white infants in the United States. So how is it possible? How is it possible, if we just talked about that there's no biological underpinning of race, how is it possible that there are differences between groups where you're using race to define those groups? So I want you to design a study to test if this difference in birth weight is due to differences in genetics. So this study has been done, and there are multiple ways that you could go about doing it, but a group of scientists, and the citation is listed at the bottom here, David and Collins, 1997. So what they did is they looked at the birth weight of babies, and they looked at the mothers, and whether the mothers were African-born blacks, U.S.-born blacks, or U.S.-born whites. And what you might notice from this graph is two of these lines are very close together, and one of those lines is different. I know it's a little bit difficult from this graph to see what's what, but it's U.S.-born blacks that you can see as that sort of line that sticks out on the left. So what would be your takeaway here? The takeaway of the authors is that the birth weight patterns of infants of African-born black women and U.S.-born white women are more closely related to one another than to the birth weights of infants of U.S.-born black women. So what their conclusion was is that this challenges the genetic concept of race as it relates to birth weight. So that the differences between the birth weight of babies that are born to U.S.-born black women versus U.S.-born white women is not genetically based. So it's not that there's not a difference, it's that that difference comes from social reasons, not from genetic reasons, which means it absolutely is something that we need to consider, we need to talk about, we need to investigate, but it is not biological in nature. The American Medical Association very recently kind of finally came to terms with this and realized that it's really important that doctors and researchers not use race in a biological way. So in November 2020, the AMA adopted policies that recognize that race is a social construct rather than a biological construct. AMA recommends that clinicians and researchers focus on genetics and biology and the experience of racism and social determinants of health when describing risk factors for disease. We're going to continue our discussion of non-Mendelian genetics, so genetics that have a slightly more complicated inheritance pattern, by looking at unlinked versus linked genes. So let's consider an individual with 3 genes of interest, and they are heterozygous for each of these. So big A, big B, and big C, little c. We're going to consider 2 possibilities. What if each of these 3 genes is on a separate chromosome, and what if all 3 genes are on 1 chromosome? And so I want you to draw each of these possibilities, assuming we're talking about a diploid individual. So in the case of the 3 genes being on separate chromosomes, we would have 3 homologous pairs of chromosomes, whereas if all 3 genes are on one chromosome, we would only need to have 1 homologous pair. Let's focus on the example of genes on the separate chromosomes first. So diagram the process of DNA replication and crossing over. So on the right, you can see what happens when we have our DNA replicated and then crossing over. So we get an exchange of information on the non-sister chromatids. I want you to list all of the gametes that can be produced by an individual with a genotype of big A, big B, little b, big C, little c, assuming that all of these are on separate chromosomes. So keeping that in mind, what's the likelihood of each type of gamete? So all of the combinations are possible, and those are listed there. And keeping in mind that they're on separate chromosomes, all of those gametes are equally likely, and that's because the chromosomes assort independently. The chromosomes containing gene A do not impact the movement of the chromosomes containing genes B and C. And if you want more information on that, you can go back to Unit 5.2, where we talked about independent assortment. Now let's consider if the genes are on the same chromosome. I want you to do the same exercise of diagram the process of DNA replication and crossing over. In this case, the location of crossing over is actually going to have an impact on what the end chromosomes look like. So if the crossover region happened somewhere in between gene A and gene B, it would look like that top possibility. And if crossing over happened between gene B and gene C, it would look like that bottom possibility. Now in real life, chromosomes are quite long, and you can have crossing over happening in multiple locations. But we're going to keep this simple and imagine it's just happening in one location. So based on this example, what determines the likelihood of each gamete that can be produced here? So in the case of the genes being on the same chromosome, not all of the gamete possibilities are going to be equally likely. Because big A, big B, and big C all started on the same chromosome, those are going to stick together unless crossing over happens in between to break them up. Same applies to little a, little b, and little c. The big takeaway here is that the closer together 2 genes are, the less often they split up, and this is known as linkage. A side note is if genes are so far apart that they separate 50% of the time, they're said to assort independently, even though they're on the same chromosome. So linked genes are genes that are so close together on the same chromosome that they are likely to be inherited together. So let's make sure you understand this by figuring out what the gamete possibilities are in this example. So what are the non-recombinant gametes? And then what are the gametes that would be a result of crossing over between A and B, and the result of crossing over between B and C? So the most likely gametes produced would be both big A, big B, big C, as well as little a, little b, little c. And those are the non-recombinant type. Please note that these stick together not because they are all dominant or because they're all recessive, but it's because they were together on the original chromosome. All right, the next most likely combination would be big A with little b, little c, or little a with big B, big C. So these gametes would be a result of crossing over between A and B. Although that's less likely than having no recombination at all, it is more likely than the case of where we had crossing over between B and C. This is least likely because those genes are so close together. So big A, big B, little c, and little a, little b, big C. So let's just summarize this and make sure that you understand the concept of linked genes. Genes are linked if they are located close enough together on the same chromosome that they tend to be inherited together. So unlinked genes are shown on the left. This is independent assortment. The genes are far apart or located on separate chromosomes. All of the gametes that can be produced will be equally likely. In the case of linked genes, these are genes that are often inherited together. So these are when genes are close together on one chromosome. For example, genes in B and C would be linked. Genes A and C probably wouldn't be linked just because they're so far apart that they would probably assort independently. But when genes are close together, like B and C, they will often be inherited together, and those are considered linked genes. Linked genes were first discovered in fruit flies, so I want to use them as an example to help you understand linked genes in a little bit more detail. So what we have is on the left, we have a female that is a double heterozygote, but displaying the dominant phenotype. On the right, we have a male that is homozygous recessive for two traits, and this is purple eyes and vestigial wings, so small wings. So he is homozygous for both of these. So what we do is we cross these individuals and then see if what the resulting offspring look like. So most of the offspring are going to get one of those two chromosomes from the female, either the one that is ++, that means wild type for both traits, or that one shown in blue that is recessive for both traits. So these are called parental because they got a non-recombined chromosome from the parent. In the case on the right, these are recombinant. So what that means is crossing over during meiosis occurred somewhere between these two genes. And so in this case, they will either display purple eyes or vestigial wings, but not both. So this happened because there's a recombination and there was a crossing over event between these two genes. Now remember that crossing over between two genes is going to be more likely the farther they are apart. So saying that another way, the closer two genes are, the less recombination you're going to have. So if we have very few recombinants, we know that those genes must be very close together. Having that information actually allows us to make a map of the genes. To make a map, we first need to figure out recombination frequency. To do that, we take the total number of individuals that were a result of recombination and divide that by the total number of individuals. See if you can do that with this example. We should end up with a recombination frequency of about 10.7%. We get that by adding up the individuals that were a result of recombination, in this case 151 plus 154 for a total of 305, and we divide that by the total number of individuals, and that should be 2,839. So when we divide those out, we get a recombination frequency in this case of 10.7%. If we do that with enough genes, we will be able to make a large map. So because recombinants happen less often when genes are close together, you can use the recombination frequency to create a gene map. So each percent of recombination frequency, like what percent of the offspring are due to a recombination event, that's considered one gene map unit. A little side note as a reminder, if the recombination frequency is less than 50%, so we get recombination less than 50% of the time, what do you assume to be true? If recombination frequency is under 50%, that's because the genes are linked, and that tells us that they are close together on the same chromosome. So in this case, we are looking at genes A, B, and C. Because the recombination frequency between all of them is under 50%, that means all of them are going to be on the same chromosome and relatively close together. We can then make a map based on these numbers. And so what this is indicating is that the largest recombination frequency tells us that those are the ones that are farthest apart. So use that information to make a map of these three genes on the chromosome. So here we go. It's as simple as that. We know that A and C are the farthest apart because they have the largest recombination frequency. And then B is in between those two. And it's closer to C than it is to A because there's a smaller chance of recombination between genes B and C. Here's just a diagram of an actual gene map of a Drosophila. So it's showing a bunch of traits that occur on chromosome 2. So we know where those traits are, or where those genes are on the chromosome because of many, many studies looking at recombination frequency between all of these genes. Final topic of this video is pedigrees. So take a look at this diagram. What is a pedigree and what do you think it's good for? So a pedigree is a way to represent the prevalence of a trait in a family, and it helps us sort of figure out inheritance pattern, and it also helps us predict the likelihood of a trait appearing in offspring. So if we look here, we have widow's peak and we have earlobe. So I want you to try to use these diagrams to see if you can figure out if each of these traits are dominant or recessive. So use that key to try to interpret these diagrams. So in the case of widow's peak, that's a dominant trait, and we can see that best because these diagrams actually give us quite a bit of information. By the picture where you see the woman who has the widow's peak, that's shown next to the genotype big W or big W little w, which implies that it is a dominant trait. On the right, we can see that attached earlobe is the recessive trait, coded for as little f, little f. So what I want you to be able to do eventually is just from looking at the shading of whether it's affected or not, be able to figure out if a trait is dominant or recessive. So for example, if we look at the two individuals in the second generation on the widow's peak, where we see that they are both affected, big W little w and big W little w, when they have kids, one of them is not affected. The only way for that to happen is if they must have been heterozygous. And so that means that no widow's peak must be the recessive trait. If you look over on the attached earlobe side, in the second generation, in the center there, we have two heterozygous individuals who do not have the trait. So two individuals who don't have the trait end up having an offspring with the trait. That means that this must be a recessive trait. So to have two parents who don't express it, have a kid who does have it, that means that that trait is recessive. So what kind of clues can we use in determining whether a trait is dominant or recessive from looking at a pedigree? So assuming it's an autosomal trait, which means it's on one of the first 22 pairs of chromosomes that humans have that have nothing to do with our biological sex, assuming it's on one of those 22 pairs, first of all, the inheritance pattern, sex doesn't matter. It's not going to be more likely in males or females. It's just equally likely regardless of biological sex. What we should look at in autosomal is the overall inheritance pattern. So for example, in autosomal dominant, if there's an affected child, there must be an affected parent. Because only one copy of the allele is necessary to display the trait, if it's showing up in the child, it must have come from one of the parents and would show up in the parent. To recognize if it is recessive, one way of looking at it is it can skip generations. Another thing to keep in mind is that you can have two unaffected parents end up with an affected child. So if you have an affected child with not an affected parent, that's a clue that this is a recessive trait. So I mentioned that we were assuming that this was an autosomal trait on one of those first 22 chromosomes. But there are traits that are on that last pair of chromosomes, which are the sex chromosomes. So in humans, females have, or biological females based on chromosomes, remember gender is complicated, but two X's results in biological female. An X and a Y results in biological male. So there are traits that are X-linked dominant or X-linked recessive, and these are traits that are found on the X chromosome and either follow a dominant or recessive inheritance pattern. An example of an X-linked trait in humans is red-green color blindness. So on the right is a circle, and if you are able to see the number 74 within that, you're probably not red-green colorblind. Where if you're unable to see that, you probably are unable to distinguish between red and green. This is more common in males than females because it's X-linked. So for females need to inherit two copies of the color blindness allele in order to display color blindness, whereas males only need to have one copy of the color blindness allele because it's on the X, and the other chromosome would be a Y chromosome. So in males, it's going to be more likely. And you can take a look at the Punnett squares shown here to better understand that. Another type of inheritance patterns in humans is Y-linked. So these are cases of genes that are on the Y chromosome. These are usually pretty easy to pick out on pedigrees because only males will be affected. There's some other ways of figuring it out. Affected males will always have affected fathers and affected sons. And again, we're talking biologically. There are lots of wonderful ways to have a family, but pedigrees are based on biological relatedness. Not very many disorders are Y-linked because the Y chromosome does not carry very many genes. But one example is Y chromosome infertility, which results in low sperm count. All right, so here is a practice problem that you can try. So here's a pedigree, and I want you to try to figure out for each of the inheritance patterns we just went over, which of these is not possible based on this pedigree, and see if you can give the explanation for why this pattern is impossible. So autosomal recessive is impossible because if it were true, two affected parents would have 100% affected kids. And you can see that in where I've listed in red the number 1. Autosomal dominant is possible. That's the most likely inheritance pattern shown in this pedigree. X-linked recessive is impossible because if it was true, the sons of affected females would all be affected. And what we see in the arrows pointing to number 2, we have sons of an affected male that are not affected. And so that makes this impossible on X-linked recessive. X-linked dominant is also impossible because if it were true, the daughters of affected males would all be affected. So that circle in the middle on the bottom right, that daughter should be affected if it's X-linked dominant and her father was affected, because she gets the X from, she gets one of those X's from her father, and he only has one X to give. Y-linked in this example is also impossible. If it was true, females would not be affected because females are XX. Now, these problems are certainly tricky. They take some time to figure out, so I highly recommend you do lots of practice problems on these to try to figure out these patterns. Once you've gone through it a number of times, they start getting easier. We're going to leave it here for today. That was a whole lot of information, so thank you for sticking with it.